Colon cancer is really preventable. We understand how it develops and we know what we can do to prevent it. By understanding how colonic cancer develops, you can be a pathology superhero. Let's first think about colonic polyps. There are several types of colonic polyps. Some are premalignant and some aren't. Colon cancer arises through the adenocarcinoma sequence. It begins with a polyp, the colonic adenoma, and ends in carcinoma. Colorectal carcinoma can also be part of inherited cancer syndromes. Let's think about some of the more innocent polyps. Hyperplastic polyps. These are really common. Most people over the age of 60 will have small hyperplastic polyps on the left side of their colon. What they look like is small nodules. People often describe them as dewdrops. They're multiple. If we look down the microscope, we can see that they are composed of mature cells, goblet cells, and absorptive cells. These cells overgrow slightly and create what we call a serrated architecture. Can you see in the bottom illustration how the normal mucosa has changed to have a kind of zigzaggy outline in this hyperplastic polyp? This is due to the overgrowth of the goblet cells. Adenomas have the potential to accumulate mutations and become carcinomas. They again are really common and they're almost always asymptomatic. They can present with occult bleeding and iron deficiency and anemia, but these days we like to detect them in the GI screening program for colonic cancer. If we look at the polyps, they can either be flat and carpet-like, like the ones illustrated in the bottom photo. These are particularly found in the rectum. Or they can be pedunculated, like in the top photo. These are found throughout the bowel. Words that we use down the microscope to describe them are tubular, meaning just using glands to form the polyp, tubulovillous, which means glands, and long frilly villi or just villi. It's the ones with just villi that have the highest premalignant potential. All the adenomatous polyps are characterized by epithelial dysplasia. Now, while most adenocarcinomas arise from polyps, most polyps will just sit around and do nothing. It's only a special few that acquire mutations that will go on to produce adenocarcinoma. In the illustrations to the right, you can see the difference between a low-grade polyp and a high-grade polyp. The high-grade polyp looks much more disorganized. There's more nuclear hyperchromasia, nuclear stratification and elongation, and much more mitotic activity. Notice as the dysplasia progresses, there's decreased numbers of goblet cells. Low or high-grade dysplasia describes how likely this polyp will turn out to be precancerous. Once again, let's think about the adenocarcinoma sequence. There are specific mutations that are acquired by normal epithelium as it journeys towards carcinoma. The first is the APC mutation. This may be familiar to you because this is the mutation in familial adenomatous polyposis. Other important mutations are the KRAS mutation and the P53 mutation. Taking non-steroidals, aspirin, can interfere with this progression of mutation. There are some other things that we can do as well. This is where the GI screening program comes in. Those polyps are just collected and taken away before they can develop more mutations. In the previous slide, I mentioned familial adenomatous polyposis. This is one of two important inherited risk factors for colonic carcinoma. The other is Lynch syndrome, also called hereditary non-polyposis colonic carcinoma. Familial adenomatous polyposis is an autosomal dominant disorder. It's due to a germline mutation in the tumour suppressor gene APC. People who carry the APC gene in one of their chromosomes can have a second hit in the other chromosome, and loss of both of these genes leads to development of an adenoma. In the case of familial adenomatous polyposis, there are literally thousands, a carpet of adenomas throughout the bowel. And each one of these has a chance of becoming carcinoma, which is why people with familial adenomatous polyposis have a 100% chance of getting colorectal cancer before the age of 30. We can't just remove individual adenomas for people with familial adenomatous polyposis. They need their entire colon removing. They also have an increased risk of cancer elsewhere in the GI tract, for example in the stomach and small bowel. This picture to the right shows the innumerable polyps that develop in familial adenomatous polyposis. Thousands coating the whole bowel. Lynch syndrome is much more common. Lynch syndrome is caused by an abnormality in DNA repair. It can be caused by a mutation in one of four genes: MLH1, MSH2, PMS2, and MSH6. Together, these four proteins form an enzyme that repairs DNA. In a specific situation, it repairs it where there has been a DNA mismatch. People with Lynch syndrome get colorectal carcinomas at a younger age. They also get cancers in other places. They get carcinomas of the endometrium, stomach, ovary, ureters, brain, and small bowel. But mostly, they get cancers of the colon, which tend to be right-sided and often mucinous. Sporadic adenocarcinoma is distributed equally over the entire length of the colon. It has different symptoms depending where it occurs. Proximal adenocarcinoma rarely causes obstruction and will often present with iron deficiency anemia. In the distal colon, an annular constriction gives rise to a tight apple core that presents with obstructive symptoms. This is a colonic adenocarcinoma. In contrast to the normal mucosa at the top right, where the glands go in a really orderly fashion, can you see those very, very badly organized glands invading underneath, spreading through the bowel wall? And the cells that line the glands are hyperchromatic, mitotically active, and piled up in an abnormal fashion. Pathologists, when they're talking about tumours, often talk about tumour stage, and bowel cancer is a good place to illustrate stage. Stage describes how far a tumour has spread through the bowel. Here we can see a T1 tumour that's confined just to the inner layers of the bowel. As it spreads through the wall of the bowel, the T stage increases. T2 into the muscularis, T3 beyond the muscularis, and T4 spread to the serosal surface. The stage will determine the outcome. This brings us to the end of thinking about adenomas and carcinomas. The adenoma-carcinoma sequence is one of the most important concepts in pathology. By knowing about this and how it relates to familial tendencies for colonic carcinoma, you can participate actively in the GI screening program and you can be a pathology superhero.